Hello, I'm Stephen Lee, and welcome to Omics Quest. Today we are diving into a quick guide on quantitative PCR, also known as real-time PCR, for measuring DNA and RNA in real time. qPCR is a versatile tool used in a wide range of applications, from accurately quantifying NGS library concentrations to testing for COVID-19. Let's break it down. qPCR uses fluorescence to measure the amount of DNA. Generally, there are two main approaches to using fluorescence. The first is cybergreen. Cybergreen is a fluorescent dye that binds to double-stranded DNA, like this. When cybergreen is free in solution, it has very low fluorescence. Upon binding to double-stranded DNA, its fluorescence increases dramatically. The second approach involves using a TechMan probe, which consists of a 5' fluorescent reporter dye and a 3' quencher molecule. When in close proximity, the quencher prevents fluorescence from the reporter dye by absorbing its energy. However, when Tech polymerase reaches the probe, its exonuclease activity cleaves the reporter dye from it, resulting in fluorescence. The remaining 3' fragment of the probe is eventually cleaved or displaced as well. Regardless of the approach used, the amount of fluorescence can be measured in each PCR cycle. Let's assume we are doing this for a sample with a known concentration of 10 to the 5th copies per microliter. Notice that it took 8 cycles to cross the threshold line. This is called the threshold cycle, or CT. We can then make serial dilutions and perform qPCR for each dilution as well, like this. Again, again, and again. We can see that the lower the concentration, the greater the number of cycles required to reach saturation. Similarly, it takes more cycles to cross the threshold line. We also need to run a negative control by performing qPCR without a template. We can then establish a relationship between the observed CT values and the DNA quantity. From this relationship, we can calculate PCR efficiency, where 100% efficiency indicates that the number of template DNA copies doubles with each PCR cycle. From this point on, absolute quantification can be performed using a standard curve, which is useful, for example, when quantifying NGS libraries prior to pooling for sequencing. Relative quantification is also possible if you're primarily interested in comparing CT values. COVID-19 testing is a prime example. By comparing the CT values of the target gene with those of a reference or housekeeping gene in both a healthy control and a patient, it is possible to determine whether the patient is positive for COVID-19. By the way, in this case, RNA must first be converted to cDNA, which is achieved through reverse transcription. Nice. Thanks for watching Omics Quest. If you enjoyed this video, please like and subscribe to the channel.